Hello, my name is Umi Alia Olia binti Salam, and I will be presenting about the introduction for the journal that our group have chosen. Organ transplantation is essential for individuals suffering from end-stage organ failure. However, there is a worldwide limited of appropriate donor organs. Xenotransplantation, namely with genetically modified pigs, has arisen as a promising treatment. The pig is considered suitable because of its similarity in organ size to human, rapid maturation, and favorable public opinion. Genetically engineered pigs, particularly those with deactivated alpha-1,3-galactosyltransferase, have the potential to decrease the likelihood of hyperacute rejection. The initial pig-to-human kidney transplants have shown positive results, with no indications of rejection. Nevertheless, conventional or traditional approaches may fail to identify initial indications of rejection or immunological response due to the limited time frame of these experiments. The problem statement of the journal is due to a significant shortage of acceptable donor organs. Innovative treatments such as xenotransplantation using genetically modified pig kidneys are required. Despite encouraging initial results, traditional approaches may miss early indicators of human response and rejection because of their short trial durations. Advanced tools like single-cell RNA sequencing are necessary to fully understand the complicated interactions between xenografts and human recipients, which will guide future genetic optimizations to enhance the safety and effectiveness of xenotransplantation.
Next, research objective. There are two research objectives from this topic. First, to analyze cellular and molecular changes in porcine to human kidney xenograft and longitudinal peripheral blood mononuclear cell (PBMC) samples using single-cell RNA sequencing to characterize cellular and molecular dynamics. Next, identify significant transcriptome alteration in human and porcine cells during xenotransplantation, specifically focusing on changes that may not be detectable using conventional histology techniques. Moving on to literature review, organ transplantation is crucial for patients with severe organ failure, but there is a shortage of donor organs. Xenotransplantation using organs from genetically modified pigs offer a solution because pigs match quickly and have organs similar to humans. In 2021, the first successful pig-to-human kidney transplant was done by removing the alpha-gal gene in pigs, which reduced rejection chances. The early result was positive, with functioning kidneys and no signs of immediate rejection. However, traditional methods may miss early signs of rejection. Advanced techniques like single-cell RNA sequencing provide deeper insight into cellular and immune responses, helping to improve future genetic modification and the overall success of xenotransplantation.
Now I'm going to present about the methodology. First and foremost is a sample preparation for bulk and single-cell RNA sequencing. Next is the data pre-processing and quality control of scRNA sequencing. After that is the data normalization, integration, and cell type annotation. Next is the differential expression analysis. After that will be the cell cycle analysis. Next will be the temporal gene set co-expression clustering analysis. After that will be the temporal enriched gene set identification. Moving on to the next is the gene set enrichment analysis for functional annotation. Lastly is the analyzing longitudinal bulk RNA sequencing of kidney biopsy samples. There are a total of 6 results obtained from the research article journal. The first one is the single-cell transcriptomic landscape of pig-to-human kidney xenotransplantation. The scRNA sequence was performed on xenografts to characterize the pig-to-human xenotransplantation associated cellular dynamics. The second result is the human immune cell infiltrate porcine xenografts. It is to test whether human immune cell had infiltrated into the xenograft. The third result will be the transcriptomic signatures indicate an antibody-mediated response. It is to distinguish different types of rejection between ABMR and acute cellular rejection. Fourth result will be the global xenograft tissue damage. It fully characterizes the cellular changes of porcine kidneys. Fifth result will be the xenograft-specific proliferating cells indicate porcine kidney tissue repair. It aims to identify a unique proliferating cell population among all kidney cell types. The last result will explain how the recipient responded to the grafting of the porcine kidney across the period of xenotransplantation.
There are three main findings from this journal. The first one is there is early signs of antibody-mediated rejection in the xenograft. The second one is there is evidence of human immune cell infiltration, endothelial activation, and tissue damage. And the third one is there is activation of porcine immune cells and proliferative cells in the xenograft. There are also a few implications stated in the journal, which are monitoring immune dynamics and cellular responses in xenotransplantation can improve outcomes. Other than that, potentially targeting tissue-resident immune cells pre-transplantation could mitigate immune responses, and last but not least, understanding the role of proliferative cells in tissue repair is crucial. There are also a few limitations, such as small sample size, short monitoring duration, and use of specific pig model, which is alpha-gal co. In conclusion, this study examines the detailed interactions between xenograft and recipients in early xenotransplantation using advanced transcriptomic methods. It highlights the advantages of these approaches in identifying rare cell populations and understanding immune responses over time. More research is needed to translate these findings into practical improvements in pig genetic engineering and xenotransplantation. That's all from our group. Thank you.